Today, we are going to learn about the reactions of the biological system. Inside all living organisms, hundreds of reactions are going on all the time with utmost precision in terms of time and space. These biological reactions, precisely controlled, have the ability to convert a small seedling to grow into a large tree or a small baby to a matured human being. Reactions of the biological system can be viewed from different points of view. In a series of 2 lectures, we will try to probe into the reactions of the biological system. In the first lecture today, we will discuss the biophysical aspects of the reactions of the biological system. And in the second lecture, we will discuss from the biochemical point of view with special reference to the specialized reactions of the biological system, namely replication, transcription, translation, and photosynthesis. In the biophysical aspects of the reaction, we will discuss about thermodynamics, the subject that deals with energy transformation between the system and the surrounding. We will also discuss about the order and molecularity of biological reactions. We will discuss about the concept of free energy and their role in determining spontaneous or non-spontaneous nature of biological reactions. Finally, we will discuss about the high energy compounds that are coupled to other biological reactions to drive any reaction which is otherwise non-spontaneous thermodynamically. The most important considerations from the biophysical point of view is thermodynamics. In thermodynamics, the organism which we are interested in is called the system. Everything else in this universe other than the system is called the surrounding. Depending on the interaction between the system and the surrounding in terms of exchange of energy and matter, systems can be defined as the isolated system, closed system, and open system. A closed system is a thermodynamic system which does not interact with its surrounding in terms of matter or energy, whereas an isolated system is one which exchanges energy with its surrounding but not matter. An open system exchanges both energy and matter with its surrounding. Now the question is, where do the biological systems stand in this regard? Biological systems are thermodynamically open systems and are far from equilibrium dissipative structures. Biological systems are called open systems because biological systems derive energy and matter from the surrounding. They also deliver or give back some amount of energy and matter to the surrounding. So in this regard, biological systems are considered as open systems. They are far from equilibrium systems because in biological system, it is never true that the amount of input energy is equal to the amount of energy given back to the surrounding. So a biological system always maintains non-equilibrium state by consuming food. Thermodynamic equilibrium in biological system means death. Biological systems are called dissipative structures because they can dissipate heat in a hierarchical fashion. In case of chemical collision theory, for a reaction to happen, 2 or more reactants must collide with each other to form a compound. So molecularity refers to the number of atoms, ions, or molecules that need to interact with each other to make a particular reaction to happen. If we think about the molecularity of biological reactions, it is really diverse. Reactions can be unimolecular, bimolecular or multimolecular depending on the number of atoms, molecules or ions that get involved in these reactions. In biological systems, denaturation of DNA, where a piece of double-stranded DNA gets denatured into two single strands, may be considered as a unimolecular reaction. But unimolecular reactions are not that frequent in biological system. Most of the reactions of the biological systems are bimolecular. The simple enzyme substrate reactions, which are present abundantly in the biological world, is a bimolecular reaction where a substrate molecule reacts with an enzyme molecule to form a product. Whereas replication, transcription, translation, these are all multimolecular reactions. Think about the translation apparatus. Ribosome provides a huge surface. Then comes the tRNA, the mRNA and the substrates, that is the amino acids, to form peptide bonds. Order of reaction with respect to a particular reactant is defined as the index or the exponent to which its concentration has to be raised in the rate equation. An example can make it clear. Suppose a reaction happens in such a way that 2A plus B equal to C. In this reaction, the reaction rate R equals K times concentration of A square times concentration of B. And the order of this reaction with respect to A is 2 and with respect to B is 1, and the overall order of this reaction is 2 plus 1 equal to 3. Now the question is, what does this order of reaction tell us? The order of reaction tells us about the relationship between concentration of the reactants and the rate of the reaction, or in other words, what will be the change in the rate of the reaction if we increase or decrease the concentration. Let us take a deep look. The rate of reaction R equals concentration to the power of n, where n is the order of the reaction in whole number 0, 1 or 2. A reaction is called a zero order reaction when the rate of the reaction is independent of the concentration. N equals 0 in case of a zero order reaction. If we plot the log concentration of the reactant at the x-axis and log rate of reaction on the y-axis, then for a zero order reaction we get a straight line parallel to the x-axis. That means the rate of reaction is independent of the concentration of the reactants. For a first order reaction where n equal to 1, the rate of the reaction is directly proportional to the concentration of the reactant. And in a similar plot where we plot at the x-axis log of concentration and at the y-axis log of rate of the reaction, we get a straight line with a slope of 1. For a second order reaction, for same type of plot, we get a straight line with a slope of 2. Now, the question is, what are the order of reactions that we see in the biological system? And the answer is, zero order kinetic reactions are rare in biological system, except for some decomposition reactions which go really slowly. Zero order reactions are not that common in biological system. First order kinetics are perhaps the most common reactions that take place in the biological world. Microbial growth and many other growth that we see in the biological world is first order kinetics. There are some examples of second order kinetics in biological reaction. The best one would be renaturation of DNA. When two single strand DNA renature to form a double stranded DNA, it can be considered as a second order reaction. Now the metabolic reactions, which are the collection of all chemical reactions that are happening inside an organism, can be classified into exergonic reactions and endergonic reactions depending on energy requirement. Now an exergonic reaction is one which gives out energy. In that case, the energy of the reactants is higher than the energy of the products. There can be an activation barrier though, but for all exergonic reactions, the amount of energy for the reactants is higher than amount of energy of the products, and there is a neat fall of delta G value or the Gibbs free energy change. For endergonic reactions or energy requiring reactions, the energy of the product is higher than the energy of the reactants, and that's why there is a neat accumulation of energy, and these reactions thus are energy demanding reactions. The catabolic reactions are reactions where large molecules break down to form smaller, simpler units, like for polysaccharides break down into monosaccharides, lipids break down into fatty acids, proteins break down into amino acids, and nucleic acids break down into nucleotides are all considered as catabolic reactions, and these catabolic reactions in the biological system are normally exergonic in nature, that is, they give out energy, whereas the reactions that build up multimeric molecules, large molecules like proteins, like nucleic acids or polysaccharides from their monomeric units are considered as anabolic reactions, and these anabolic reactions most of the time are energy demanding or endergonic reactions. There are some reactions that are categorized as amphibolic reactions or a mixture of both catabolic and anabolic reactions. And because of these dual metabolic roles of these reactions, they are so named the amphibolic reactions. A good example of amphibolic reactions are the first few reactions of the TCA cycle or the citric acid cycle. The very first reaction of citric acid cycle is where a 4-carbon compound oxaloacetate gets joined up with acetyl-CoA, a 2-carbon molecule, to form citric acid, a 6-carbon compound. This is an example of an anabolic reaction. In the next few steps of the TCA cycle, the citric acid gets rearranged to form isocitrate, which eventually breaks down into succinate, which is a 4-carbon compound, and 2 molecules of carbon dioxide is released. So, breaking down of isocitrate to succinate and carbon dioxide is considered as a catabolic reaction. So, the first few reactions of the citric acid cycle is both catabolic and anabolic, and that's why these reactions are called amphibolic reactions. Gibbs free energy, as it is called in the IUPAC, Gibbs energy or Gibbs function, is a thermodynamic potential which measures the useful work that can be obtained from a thermodynamic system. In biological systems, entropy and energy, they change simultaneously, and Gibbs free energy is that part of energy which can be used for downstream reactions. The entropy increases for all biological systems. The concept of Gibbs free energy can be discussed for a reaction where A plus B reversibly forms C plus D. The change in Gibbs free energy delta G equals delta G 0 plus log of concentration of C times concentration of D divided by concentration of A times concentration of B, where delta G0 is called the standard free energy change of a particular reaction. Now this value of delta G for any biological reaction is of extreme importance. What does the value of delta G tell us? The spontaneity of a biological reaction. It tells us whether a reaction is spontaneous to be happened in nature or it needs some extraneous help. If the value of delta G comes negative, then the reaction will be spontaneous thermodynamically, and if the value of delta G is positive, then the reaction is non-spontaneous. Coupled reactions in biological system are extremely useful. Many a times we see that a reaction which is otherwise thermodynamically not spontaneous but still can happen in nature with the help of getting coupled with another reaction. A good example is glucose and fructose joining together to form sucrose. The delta G value of this reaction is plus 5.5 kilocalorie per mole. That means the delta G value is positive, meaning this reaction is not spontaneous in nature. However, another reaction where ATP breaks down to form ADP and PI has a delta G value of minus 7.3 kilocalorie per mole, which is highly spontaneous. Now, if these two reactions can be coupled, then with the help of breakdown of ATP, formation of sucrose from glucose and fructose is possible. And the reaction goes like this. Glucose binds to one molecule of ATP to form glucose 1-phosphate and ATP. And this glucose 1-phosphate then binds with fructose to form sucrose and PI. And if you look at the overall reaction, it becomes glucose plus fructose plus ATP becomes sucrose plus ADP plus PI. And the net change of free energy, that is delta G, is minus 7.3 plus 5.5, which is minus 1.8. That means when these two reactions are coupled, the change of delta G is minus 1.8 kilocalorie per mole, and the reaction of formation of sucrose from glucose plus fructose when coupled with hydrolysis of ATP becomes spontaneous in nature. There are some compounds called the high energy compounds, like we have just seen the ATP molecules. These high energy compounds are of extreme importance in the reactions of biological system. They have the potential to pull reactions that are otherwise thermodynamically not spontaneous. ATP is a cardinal such molecule known as the energy currency in the biological system. Hydrolysis of GTP can also be of importance as it is used for protein synthesis and involved in many signal transduction process. These compounds are extremely helpful to drag reactions that are otherwise thermodynamically not spontaneous. We conclude by saying that biological systems may be looked at as a summation of hundreds of reactions that are going on in a precise and controlled way. Thermodynamically, biological systems are open systems, far from equilibrium, dissipative structures. Most of the biological reactions obey first and second order kinetics and with higher values of molecularity. A vast majority of the biological reactions may be thermodynamically unfavorable, but still they happen in nature because they get coupled with some reactions that are thermodynamically much more feasible. These coupled reactions are of extreme importance in biological system. High energy compounds like ATP, GTP and other phosphate anhydrides are present in abundance in the biological system to drag reactions that are otherwise thermodynamically not spontaneous. Catabolic reactions are mostly exergonic in nature, meaning they give out energy, and anabolic reactions are mostly energy absorbing reactions as they build up the body of the organism's systems and organs.